Hello everyone. So today we are going to talk about oxygen dissociation curve in particular, and a lot of students are requesting to make this a tutorial video for this particular topic because a lot of students, I don't know why, but they are not able to understand this topic particularly, and they face a lot of difficulty to understand this topic, that what is happening, where is happening, and also since the understanding the fundamentals is not clear, they're not able to solve a lot of questions based on this topic. And this one is an important topic, you can say. A lot of competitive exams like IGSO or KVPY, even NEET, they ask numericals or the questions based directly on this concept, oxygen dissociation curve. So today I am going to try a method that will make this whole topic a bit easy for you to understand. So I'm sure by the end of this lecture or topic discussion, you will be able to understand the whole topic. And so just let's begin, and do comment and let me know whether you have understood this topic or not. And so let us begin. So oxygen dissociation curve. So in order to understand the whole topic, the main topic, that is oxygen dissociation curve, you should understand some basic things or the history about it, right? uh, Whatever the main thing is, whatever the main problem in our day-to-day life as well, if you don't know the history, you cannot judge that particular situation, right? If any situation is in front of you, you need to know all the basics and histories first about the history of of all that situation, and then only you will be able to understand this that particular situation like easily. So let me make you understand the history first, and then we will come to the main story. Okay, so history goes like this, that the history is all about what is happening in lungs and what is happening in tissues. Okay, so here is our lungs and tissues. So what happens, there is always gaseous exchange happening within our lungs as well as tissues. Whenever we are breathing in, the breathing out, the respiration is happening. So gaseous exchange is happening in both these sides, right? In lungs as well as tissues. So what is happening, lungs, whenever we breathe in, what our lungs does, they send oxygen, a lot of oxygen to our tissues, right? So they are sending a lot of oxygen to our tissues, where respiration, cellular respiration will be happening, and ultimately our tissues are getting enough energy, right? So now once the oxygen is delivered to the tissues, what is happening within the tissues, the tissues are, they are sending back a lot of carbon dioxide. So I can write like this, they're sending back a lot of carbon dioxide, right? So oxygen, it is being delivered by lungs to the tissues, and tissues are sending back the carbon dioxide to the lungs. So this is what is happening in these two organs. Now transport, if I talk about transport of both these things, transport of oxygen, it takes place in two forms. One is in the form of oxyhemoglobin, that is bound with hemoglobin. Oxygen molecule, they are bound with hemoglobin, so it is known as oxyhemoglobin. While another form is, it can also travel in the form of, in dissolved form, in plasma. So around 97%, that is majority of our oxygen, it is being traveled in oxyhemoglobin form, but rest 3% can also travel through dissolved form in plasma. So these are two ways in which the oxygen it travels throughout our body. Now if I talk about carbon dioxide, let us talk about carbon dioxide now. So carbon dioxide can travel in three ways, and these three ways are, the very first form is dissolved form in plasma. So let me just write it, dissolved form, of course, in plasma. Then another form is your protonated hemoglobin. It can go with protonated hemoglobin. And here what happens, some amount of bicarbonate ions, they remain suspended in plasma, so that what happens, once this protonated form of hemoglobin, it is inside the lungs, this hemoglobin is going to get released in order to catch oxygen. So this proton then combines with this bicarbonate ions and ultimately will give you carbon dioxide plus water in the presence of an enzyme, that is carbonic anhydrase enzyme. Then it can also travel in bound form with carbon dioxide, right? So these are the three forms in which CO2 can travel. Now what is happening, why I'm telling you all this, that by considering all these points, we can say that oxygen, it travels in the form of oxygen, like oxyhemoglobin. Then what happens, if it comes in contact with high proton area where the proton are very much high, then what happens, this oxyhemoglobin, it is going to get breakdown and ultimately this proton will bound to this hemoglobin and oxygen will be released. Okay, and same thing will happen, this oxyhemoglobin when comes in contact with carbon dioxide, again this hemoglobin is going to bond with this carbon dioxide and will give you oxygen. Okay, so basically what we are noting here, we can note that in presence of high proton and high carbon dioxide area, in presence of in area where these protons and carbon dioxide are present in higher amount, ultimately the oxygen will be released from the bound form of hemoglobin, right? This thing we need to remember in order to understand the further concept. So by looking at all these conditions, can I say that carbon dioxide or carbon dioxide or proton are affecting the affinity of the hemoglobin for oxygen, right? Carbon dioxide and proton, they are affecting the affinity of the hemoglobin for oxygen, and this thing is known as your Bohr effect. Bohr effect, we will use it in further concept. Okay, now if opposite will happen, let's say here oxygen, presence of oxygen, higher of oxygen, if in any area the oxygen is present in high amount, then presence of this oxygen, it is also going to affect, so oxygen is affecting the affinity of hemoglobin for carbon dioxide and proton, then this thing is known as Haldane effect. So we will use it and I'll make you understand what is actually happening. But this thing is clear that carbon dioxide and protons, if they are present in higher amount, then it will, and the oxyhemoglobin will just break down and ultimately will give you oxygen molecule will be released out of it. So carbon dioxide and proton will be affecting the affinity of hemoglobin for oxygen, while oxygen is affecting the affinity of hemoglobin for carbon dioxide and proton. This one is known as Bohr effect, while other one is known as Haldane effect. So this is the one background or one history that you need to remember. Let us move on to another thing that you need to remember. Another thing is that it is about hemoglobin. Hemoglobin, it is a respiratory pigment in our blood, right? So what you need to remember, you need to remember some facts about hemoglobin, that around 12 to 16 gram, approximately around 12 to 16 gram of hemoglobin is present per 100 ml of blood. Okay, and also 1 gram of hemoglobin, 1 gram of hemoglobin, it contains around 1.34 ml of oxygen, around 1.34 ml of oxygen. So can I say that 15 grams of hemoglobin, 15 grams of hemoglobin, it is going to contain 15 into 1.34, that will come around approximately 20 ml of oxygen, 20 ml of oxygen. Therefore another fact that you need to remember, therefore that I can say that 100 ml of blood is going to have 20 ml of oxygen, right? Because see, per 100 ml of blood we are having approximately 15 gram of hemoglobin. So 15 gram of hemoglobin is having 20 ml of oxygen. So approximately around 100 ml of blood, per 100 ml of blood we have 20 ml of oxygen. Another fact that you need to remember. Okay, now let us start with the main story. So the main story is about your oxygen dissociation curve. So what is oxygen dissociation curve? Okay, so in order to understand that, first remember that when we are sitting and we are not doing a lot of things, we are at rest, our body is at rest, then requirement of oxygen will be low. Of course, requirement of oxygen will be low, right? But when we are doing strenuous exercise, we are doing a lot of heavy exercise, then requirement of oxygen will automatically increase in our body, right? So can I say that during a particular situation in our body we require different amount of oxygen, so during which particular situation in our body how much oxygen is given to the tissues is what is oxygen dissociation curve. I'm repeating it. During which particular situation how much oxygen is given to our tissues in our body, this is what is oxygen dissociation curve. In which particular situation how much oxygen is needed, how much oxygen is required, how much oxygen is given to the tissues in our body, right? It depends or it varies situation to situation. Now this is what is oxygen dissociation curve. In order to understand oxygen dissociation curve, you should understand something known as partial pressure as well. So what is partial pressure? Partial pressure is pressure of an individual, pressure of an individual gas in a mixture of gases, This is what is partial pressure for a particular gas. For a particular gas, the partial pressure will be pressure of an individual gas in a mixture of gases. So if I require partial pressure of oxygen, I should know that how much pressure of oxygen is there in mixture of or in presence of other gases. So can I say that if partial pressure of oxygen is high, then formation of oxyhemoglobin will also be high? Of course, because in presence of high amount of oxygen, the hemoglobin will catch a lot of oxygen, right? Its affinity will be towards oxygen. So partial pressure is pressure of an individual gas in a mixture of gases, and if the partial pressure of oxygen is high, the formation of hemoglobin will also be high. Now let us understand this graph that is about partial pressure of oxygen and percentage saturation of hemoglobin with oxygen, in which a lot of oxygen in which a lot of students, they do a lot of mistakes and they remain confused. So let us take away all the confusions today. Okay, so in order to understand this curve, first understand that this at y-axis, this is the y-axis and this is the x-axis, of course, right? So on x-axis we have partial pressure of oxygen, it is increasing per mmHg of blood, and here what is happening, percentage saturation of hemoglobin with oxygen is increasing, like 20, 40, 60, 80 and 100%, right? So y-axis, what y-axis is telling us, y-axis is telling us that simply it is telling us percent saturation percent saturation of oxygen with hemoglobin. It is telling us percent saturation of oxygen with hemoglobin. Okay, now what happens, we know that one molecule of hemoglobin, it maximally can bind with 4 molecules of oxygen. So 1, 2, 3 and 4. Maximally one hemoglobin molecule can bind with 4 molecules of oxygen. So now, in order to understand and make make this graph simple, just remember a simple formula. That percent saturation of oxygen with hemoglobin is equal to number of oxygen molecule attached with hemoglobin. So number of oxygen molecule that are attached with hemoglobin divided by total number of oxygen molecule that are bound with, or that can bound with hemoglobin. So bound with hemoglobin, and just multiply all these things with 100. Just multiply with it with 100 in order to convert it into percentage, of course. So therefore I can say, if at any time only one molecule of let's say one molecule of oxygen is bound with hemoglobin, but it can see number of oxygen attached with hemoglobin is 1, so total number of oxygen bound with hemoglobin, or maximum number of oxygen molecule that can bound with hemoglobin, will always remain 4. So 1 by 4 into 100 will give you 25%. So when one oxygen molecule is attached with hemoglobin at that particular time, 25% of hemoglobin is saturated. 25% of hemoglobin is saturated with oxygen, right? In the same way, if two molecules of oxygen are attached with hemoglobin, that will give you 50% of hemoglobin that is saturated, right? So by like using this particular formula, you can calculate percentage saturation of oxygen with hemoglobin. This is one thing about y-axis. Okay, now another thing, yes, by using this all thing, you can just calculate percentage saturation of oxygen with hemoglobin. Like here, let's say this is 0 to 20, then here somewhat you will be finding 25%. So at 25%, you will be having one oxygen molecule attached, right? Then at if I talk about 50%, you will be having two oxygen molecule attached, right? Then again at 75%, which will come around here, you will be having three molecules of oxygen attached with hemoglobin. And when the hemoglobin is 100% saturated, then there has to have four molecules of oxygen attached with hemoglobin, right? So this is what is percentage saturation oxygen with hemoglobin. Now, by using all these, we will find out, and we will use different things in order to calculate that at what particular situation, in what particular situation, how much oxygen can be delivered to tissues and how much will remain in our blood. Let us understand that part now. So let me take the very first coordinate here, where our 100% our hemoglobin is 100% saturated with oxygen. So here at this particular point, our hemoglobin is 100% saturated with the oxygen. Okay, so this will come at around, this is your A point, let's say, and this will come at around 98 mm of Hg oxygen, partial pressure, when partial pressure of oxygen is around 98 mm of Hg, right? So I can say at particular A point, let me just write the coordinate for this. So for A point, what will be the coordinates? The coordinates will be, they will be 98 at x-axis plus 100 at y-axis. So 100% पे you can say the at the partial pressure, when the partial pressure of oxygen is around 98 mm Hg, the hemoglobin is 100% saturation, and this condition, it occurs in lungs. Vacha, where the oxygen will be very much high, so partial pressure of oxygen will be very much high, and ultimately the hemoglobin will be 100% saturated. Okay, now another situation B. So situation B will, when 75% of hemoglobin is saturated. So 75% will come somewhat here, right? 75%. So it will give you, the graph will drop down at x-axis on 40, like somewhat around here. So this will be your B coordinate now. So for B, the coordinates will be 40, partial pressure of oxygen will be 40, and here 75% of hemoglobin is saturated with oxygen. Okay, and this condition remains in our body when we are just sitting and we are just resting. So when our body is at physiological rest, at that time we don't need a lot of oxygen, but only that much of oxygen is required, that is needed in order to survive. Okay, so this condition is when you are just resting, you are just watching a movie, having popcorns, just sitting and lying. Okay, so this is physiological rest condition. So in this particular condition, bacha, what happens 75% of oxygen, it remains as oxyhemoglobin in the blood. Okay, and rest only 25% of oxygen will be given to tissues in order to do cellular respiration and ultimately provide some energy. Okay, so let's say at any situation, like at any like in any situation, let's say if 20, let's say in 100 ml, we have already done that in 100 ml of blood, per 100 ml of blood 20 ml of oxygen is present. So let's say if per 100 ml of blood 20 ml of oxygen is present, so in this particular situation, when our body is at physiological rest, in this particular situation, from this 20 ml of oxygen, 75% is present in the blood in the form of oxyhemoglobin. So I can say 15 ml is will remain in our blood only. 15 ml will remain in our blood only, and only from this 20 ml of oxygen, 5 ml of oxygen is delivered to tissues, right? So whenever our body is at physiological rest, you need to remember 75% and 25%. 75% will remain in blood, while 25% will be provided to tissues. Okay, now let us talk about another situations now. Let's say now we have started with, like we have started a bit of exercising. Okay, I'm doing a little bit of exercise, you're doing mild exercise, a simple yoga or something. So at that particular point, let's say we take it this situation as C. At this particular time, bacha, your 50%, your 50% of hemoglobin, 50% will come here. So 50% of hemoglobin will remain saturated with oxygen, and during this happens when the oxygen partial pressure is around, it will be around 50. So here 20, and it will come at around here like 50, approximately 50. Okay, so the coordinates for C will be, do it like this. Sorry, it will come around 30. So it will be, this is 20, 30 and 40, right? So it will come around 30 partial pressure, when the partial pressure of oxygen is 30 mm per Hg, and 30 mm Hg, and at that particular situation, 50% of hemoglobin, 50% of hemoglobin will be saturated with oxygen, and this happens during mild exercise, when you are doing a bit of activity. So mild exercise, when a bit of activity is happening. So in this particular situation, when I'm saying the hemoglobin is saturated with 50% of oxygen, 50% of oxygen will remain in blood, again in the form of oxyhemoglobin, while rest of the 50% is going to be provided to tissues in order to perform cellular respiration and in order to provide you some energy, you know? So what is happening here? Let's say again, in 100 ml of blood, 20 ml of oxygen is present. So from this 20 ml, 50% will remain in blood, that is 10 ml will remain in your blood, while rest 10 ml will be given to your tissues, right? So this is what is happening when you are doing a bit of activity, or you're doing just mild exercise, right? Now another situation can be, it can be when you are doing a lot of exercise, you are running, you are doing strenuous exercise, heavy exercises, and a lot of activity is happening. So at that time, when your body is doing a lot of activity, at that particular time, 25% will come here somewhat. So 25% of hemoglobin will remain saturated with oxygen, and it will be happening when the partial pressure of oxygen is around 20. So it will come here, 20, right? So for D, I can write, this was C, this is D. So for D, we can write the coordinates. The coordinates will be 20. Partial pressure of oxygen is 20, and at that partial pressure, 25% of hemoglobin will remain saturated with oxygen. And this condition happens when we are doing a lot of strenuous exercise, okay, a very heavy exercise is happening. So again, 25%, so 25% of hemoglobin is saturated with oxygen. So this 25% will remain where? In blood, while rest 75% of oxygen will be provided to your tissues. A lot of oxygen is being provided to the tissues because we are doing strenuous exercise, heavy exercise, and our body require that much of oxygen, right? So again, if in that particular situation, per 100 of ml of blood is having 20 ml of oxygen, so in that condition, in your blood, only 5 ml of oxygen will remain. Rest 15 ml from this 20 ml, rest 15 ml of oxygen will be given to your tissues, right? So this is about 4 conditions when and how the partial pressure of oxygen, it changes provided with different situation, and along with this, the percent saturation of hemoglobin with oxygen will also change, right? So this is about, or this is all about your partial pressure of oxygen and oxygen dissociation curve. Now only one small thing is left, that is the Bohr's effect and the Haldane effect. So what happens, let us just quickly go over it and you will be able to understand. So what is happening, this is in the normal situation, this is happening, that this is the normal curve that we are having, but like providing different situation, if the situation varies in our body, so according to that situation, this curve can either shift to a bit left or it can shift to a bit right, okay? So if it is shifting to a bit left, let's say the curve is going like this, then this left, this is known as left shift, this is known as left shift, okay? And if the curve, it is coming a bit right, according to a particular situation, it is coming a bit right, then it is known as the right shift, So when it happens, when is it happening? Left shift and right shift, why is it happening? When is it happening? So let us understand that. And in order to remember this, just remember whatever I'm going to provide you here, and you will be able to solve any question based on this, okay? So when I'm talking about the right shift, then at that particular situation related with Bohr effect, I'm talking about Bohr effect, okay? So how will you remember that this one is right shift is Bohr effect? This R is here, this R is here. So remember, during right shift, Bohr effect is getting applicable. So during this condition, whenever right shift is happening, what is going to happen? When all this right shift is going to happen, what situation will be? It is going to happen when oxyhemoglobin is breaking down. When oxyhemoglobin, it is breaking down. So breakdown of oxyhemoglobin is happening, okay? So breakdown of oxyhemoglobin, where does it happen? It happens in tissue. It happens in tissues only, in order to provide this oxygen to the tissues for cellular respiration. So the site will be your tissue. The site will be tissue, okay? So in your tissues, partial pressure of oxygen will remain low. It will remain low because their oxygen is getting delivered to the tissues, okay? The partial pressure of oxygen is low. Then temperature will remain high. Temperature will remain high. Why? Because a lot of metabolic activities is happening, so temperature will remain high. Okay. Then protons, also your protons will remain high in your tissues, and since the protons are remaining high, the acidity will also remain high. So I can write the pH will be low. When protons are high, the acidity will remain high, so pH will remain low, right? And also the bisphosphoglyceric acid, or bisphosphoglyceric acid, it is a product that is happening because of glycolysis formation. Glycolysis is happening in cell, so also bisphosphoglycerate condition is high. So when all these conditions are applicable or happening in your tissues, this particular graph will shift to a little right, towards a little right, and this is known as your Bohr effect. Okay. Now if I talk about another thing, that is your left shift. In which particular situation the left shift will happen? So left shift will happen during whenever Haldane effect is happening. So Haldane effect is applicable. So left shift will happen during what situation? Here everything will be opposite to what we have studied during Bohr effect. Okay, so here in the case of when Bohr effect is applicable, when there is breakdown of hemoglobin or oxyhemoglobin is happening, here the oxyhemoglobin will be forming. Oxyhemoglobin formation will be taking place, and this oxyhemoglobin will formation will takes place where? In your lungs. In your lungs, because whenever we breathe in a lot of oxygen, it enters inside our lungs, so the partial pressure of oxygen within our lungs will remain high, and so hemoglobin will catch up those oxygen and it will form oxyhemoglobin. Okay, so the site will be lungs, and within your lungs your partial pressure of oxygen will remain high. This is going to happen when temperature will be low. Opposite things will happen. Okay, then your protons here, the protons, it will lessen number. If the protons are less in number, the pH will be high, so the condition will be alkaline, not acidic. And also here in your lungs, no glycolysis is happening as such, so base phosphoglyceric acid or the bisphosphoglycerate, the concentration of this will also remain low. So all these situation can be asked in a way of question, that in which particular condition or in which particular situation whether Bohr effect is happening or the Haldane effect is happening. Or they can ask you a question in such a way that Bohr effect is applicable in high pressure of oxygen, or low pressure partial pressure of oxygen, or high acidity or low acidity, or high temperature, low temperature. So they will give you number of situation, and you will have to find out whether the Bohr effect is happening or the Haldane effect is happening. So I'm sure with all these things, the oxygen dissociation curve concept is clear to you, and I have tried to make it a bit easier for you to understand, and just try out a lot of question based on this topic and see whether you are able to do or not. Okay, and let us just do one question on this, so that you will be able to have an idea what particular type of question can be asked from this topic. So here the question is, how much of oxygen is delivered to the tissues during physiological rest condition by 1 liter of blood? The question is asking how much of oxygen is delivered to tissues during physiological rest condition, when we are just sitting and relaxing, by 1 liters of blood. 1 liters of blood. So if you will just remember, we have just discussed that per 100 ml of blood, it has 20 ml of oxygen. Okay, per 100 ml of blood it has 20 ml of oxygen. So can I say therefore 1 liters of blood will have around 200 ml of oxygen. Simple mass, right? So from this 200 ml of oxygen, we have just seen that at physiological rest condition, let me show you again here. At physiological rest condition, 75% of oxygen remains in our blood, while 25% of oxygen is given to tissues, only 25%. So here also just apply this thing. We have given with in 1 liters of blood, we know, we have just calculated that 200 ml of oxygen is present. So from this 200 ml of oxygen, 75% will go or will remain in our blood, while only 25% will be given to our tissue, because only that much is needed when our body is at physiological rest. Okay, so we have 200 ml of oxygen, so 75%, that is your 150 ml. 200 ml, 75% of 200 ml will be 150 ml. So this will remain in your blood, while rest 50 ml will be given to your tissue in order to provide or do cellular respiration to provide energy. So this will be your answer, that how much oxygen is provided to tissues during physiological rest condition by 1 liters of blood. Okay, so 50 ml is your answer. So I hope this topic is clear to you now. Do a lot of question based on this and let me know whether you have understood this topic or not. And I'll see you during next video. So stay tuned and bye bye.